name is CJ Norsigian, and I'm a fourth-year PhD student in the Systems Biology Research Group, and my research interests lie in using comparative genomics to find key differentiators between different strains of bacteria that are driving questions involving antimicrobial resistance and other interesting things like metabolic engineering.
Hi, my name is Xin Fang, and then I'm currently a fifth-year PhD student in the lab. My research interests involve studying the gut microbiome of inflammatory bowel disease patients, as well as using genome-scale models and other systems biology approaches to answer biological questions.
We were motivated to enumerate the protocol for making multi-strain genome-scale models because at a variety of conferences, this was a technique we were employing, and we were presenting this to other researchers. They were really interested in how we were taking a base model and using it to make predictions about other related strains. So there was a pretty obvious need and interest for analyzing these kinds of strains in this way, and so we decided to enumerate the steps and pair it with the original protocol for making a base model as a natural extension and sort of new use cases as numbers of sequences continue to grow.
So in this protocol extension, we created this new protocol that allow users to actually generate strain-specific models from a well-curated reference model. And this protocol will allow people to actually use semi-automatic notebooks to accelerate the generation of these models. The users can refer to the supplement for the Jupyter Notebook tutorial we have created to guide the users to accelerate this progress. The protocol can be broken down into four main stages. The first stage, as shown in the figure here, is to actually generate or get the base reference strain. If the reference strain is not available, one can create this from scratch following the original protocol. The second stage involved the genome mapping between the target strains and the reference strains. And the output of this stage would actually be the homology matrix that tells you the presence and absence of the reference genes within the model. The third stage involved creating the draft models from the output of the second stage, which involved deleting the genes and reactions that are missing in strain-specific models and updating GPRs. And the last stage, which we're not showing here, is stage 4, is the manual curation of the strain-specific models, which the users can follow the original protocols for quality check and refining the model, as well as adding in a new metabolic reactions and genes that are absent from the original model. And from then on, people can use these strain-specific models for a wide variety of applications.
So the main challenge for us when writing this protocol paper, in that it is a step-by-step instruction of how to execute this technique, was to try and write it in a way that would be easily accessible for people with varying levels of computational expertise or varying levels of microbiology expertise, and try to make it a way that can hit the broadest audience while also being concrete and in-depth enough for everyone to use. So we hope that the Jupyter Notebook tutorials aid people who may have a little less computational expertise, but also provide an avenue to even include some more specific things from a particular organism of interest as well. The overall core value of producing this protocol is to increase the scale of strains that are readily accessible for analysis by researchers when using genome-scale metabolic models. So by increasing the number of strains, it opens up the possibility to do different types of analyses with all of those strains in parallel. At the order of tens of strains, you can look into different growth capabilities of those strains with these draft-specific models, and then with additional curation and additional strains, you can work your way up towards even doing global epidemiology and strain tracking.
And so far, there have already been multiple studies that utilize the approaches we described in this protocol to study various species, including E. coli, Salmonella,
Acinetobacter baumannii,
and as well as many other species. So we expect that by publishing these protocols, we would enable more people to utilize this approach to study other species as well as pathogenic strains to actually deepen your understanding of the pan-metabolic capability as well as the strain-specific differences across species. I think there are a wide variety of application in health, disease application, as well as neuromicrobiology understanding.